So Anna is a postdoc in the University of Rijeka, Croatia.
Yes, correct, correct. That would be Rijeka in Croatian. I'm sorry to interfere with your proper pronunciation.
But that's all right. And she'll be talking about influence of dopamine signaling and oxidative metabolism on fluorescent advanced glycation and products formation in Drosophila melanogaster.
Yeah. Thank you, Adele, for a nice introduction. And yeah, I will start talking about one invertebrate model organism, which is Drosophila melanogaster, commonly known fruit fly. And as you actually said, I work at the Department of Biotechnology at University of Rijeka in the laboratory of Razi Andretić-Poldovsky. So our focus in work is basically associated with the drug addiction and also implication which it has on the brain plasticity and induced changes. So this work is one small side project or piece of what we actually doing. So I will start with the brain energy metabolism and dopamine implying age formation. So we all know that our CNS or brain is feeding by glucoses, which can be delivered through the GLUT3 transporter on the membrane of the neurons. But also we know that we have energy shuffling due to the lactate production inside the astrocytes, which then can feed our neurons. So what I want to emphasize now is that we know that pharmacological manipulation of dopaminergic system can affect glucose homeostasis in our neurons. And also I want to emphasize that if we have a situation with the increased amount of the glucoses, which is induced by hyperglycemia, then we have something that calls age, which are advanced glycation end products, which are non-enzymatic glycation of the protein structures, process which is reversible post-translation modification of the protein structure. And it is also really important to emphasize that AGEs are a really heterogeneous group of compounds, and they are implicated in diverse metabolic changes and also some neurodegenerative disease. But there is, since now there is no clear connection between glucose metabolic deficiency and phage formation. So now I will explain to you what phages are. So AGE are heterogeneous group of compounds, so that are all the extracellular and intracellular protein modification induced by non-enzymatic addition of covalent addition of sugars on their structures. Phage have tendency to aggregate, but they are separated in 3 different groups, and the group that we are interested in are those with the autofluorescence properties. This is important because it is allowing us accuracy, selectivity, and sensitivity in the quantification. And the most important of all, it already has some clinical application in terms of the determination of the phage in patient skin. So this was the reason why we focus all of our work on the phage formation. So our aims in our work was to synthesize and characterize phAGES-BSA standard calibrator, then to define cellular localization of phage in our model organism, also to test the influence of monoamines on phage formation in vivo, then see how does short and long-term imbalance in DNA signaling and phage formation is induced in vivo, and then to see if we can somehow inhibit phage formation by applying antioxidants. So to synthesize our standard calibrator, we use our model protein, which is BSA or bovine serum albumin, together with the reductive sugar glucoses. And then we heat it up on the 50 Celsius degree in the dark, and that process was up to 96 hours. So this is important since we were able to synthesize fluorescent advanced glycation end product of the model protein BSA. Since we know that this process of glycation is really slow, heating our samples on the 50 Celsius degree was success, and we were able to get it the phAGE-BSA final product after only 96 hours. So we wanted to characterize our standard calibrator by using emission fluorescence spectroscopy, from which we can see that only samples containing fAGES-BSA were having a maximum of emission fluorescence at 440 nanometers, which is characteristic for phage in the samples. In order to see if there is correlation in concentration regarding relative fluorescence intensity, we saw that we have a linear correlation and concluded that fAGES-BSA can be used as a standard calibrator. In order to test where the phages are inside of the tissue samples, we actually took 5 adult male flies in each sample and put them in the empty tube while we did the mechanical homogenization. By applying PBT protocol, which means that we apply buffer plus non-ionic detergent, we were able to extract extracellular and cytosolic proteins. Then we test protocol with the trypsin proteolysis in order to digest extracellular and cytosolic proteins, and also only PBS in order to focus ourselves only on the extracellular proteins. Regarding the cellular localization of phage formation, we have situation when we have excess of glucose, all-methyl glyoxylate, which is the final product of the glycosylation reaction. And this is important to emphasize since this product over here is 50,000 times more reactive than this glucose over here in order to add itself covalently, but also non-enzymatic to protein structure, which are in the extracellular or intracellular space of the cell. So what happened is that we have then intracellular and extracellular phage formation, which is then implying different protein function changes and also signal cascade and gene expression. So what we have seen is that when we applied only a PBT protocol, which was extracting both extracellular and intracellular proteins, we were seeing that that is a actually product of intracellular proteins, since the PBS protocol had really low amount of the phage in the samples. But when we look into the phage concentration and influence of the trypsin digestion, we saw that there is no influence on the protein concentration regarding phage concentration. So we actually decided to focus ourselves on the intracellular and intercellular phages, since we know that DA transports, so dopamine transport and signaling influence pre- and postsynaptic neurons together with synaptic cleft proteins. Then we tested the influence of the playing around with the dopamine concentration. So I know that this is not the case with the vertebrate models, but in invertebrate models like Drosophila, feeding flies for 48 hours with triiodotyrosine, which is inhibitor of dopamine synthesis inside of the neurons, or L-DOPA, which is a precursor of the dopamine synthesis, we can increase or decrease amount of the dopamine inside of Drosophila whole body. So what we saw is that when we reduce or when we increase amount of the dopamine inside of the vesicula, or let's say better increasing overall DA pools, we don't have any influence on the phage formation in vivo. Based on that, we tried to look into the imbalance in the DA transport and signaling, and then how that is actually influencing phage formation. And what we did did the same protocol as it was said before. We fed flies with the methamphetamine or cocaine, which are known psychostimulants affecting the dopaminergic system. And also we used 2 mutant fly lines, which are fumin with the mutation in dopamine transporter and dump with the mutation in dopamine receptor. And what we can see from this data over here is that short-term manipulation and influence on imbalance in DA transport and signaling impairment is less pronounced than the long-term impairment in DA transport and signaling induced in mutant flies. In order to better understand what is happening in METH and cocaine treated flies regarding increased amount of the phage in METH group compared to the cocaine, we can see that we have increased the amount of the dopamine in the synaptic cleft when we apply both of the drugs. But major difference is looking at the mechanistic level of the action of those two drugs is that in the methamphetamine case, we have increase of the free dopamine inside of the neuron. So we have huger efflux of the dopamine in case of the methamphetamine compared to cocaine. When we look into the fulmin and dump mutant, so this is the mutant in dopamine transporter, this is the mutant on dopamine receptor, we again have a different situation. We have change in the amount of the dopamine. So this one is synaptic and this one is extracellular. And when we look into the abundance of the phage and compare fulmin to dump mutant, we again can see that there is increase in dump mutant compared to the fulmin. And we actually believe that that is the case because we again have more of the free dopamine inside of the cell. And as we showed earlier, that intracellular phage are more pronounced the total phage when we do whole body extraction of the protein. And then to conclude this part of the dopamine impairment, we can see that both cases, when we have increased amount of the dopamine inside of the cells, we have more of the phage formation. Nevertheless, is that short or long-term manipulation with the DA transport and signaling. And then we can conclude that phage concentration in vivo is independent of overall DA pools, so those vesicular pools, but influenced by DA impairment in transport and signaling. And finally, what we wanted to test, if we can inhibit this process of phage formation induced by methamphetamine free dopamine oxidation. So we used quercetin, which is known inhibitor for phage in vitro and in vivo. And what we saw that quercetin alone does not influence phage formation in vivo that is significant. But when we apply methamphetamine and quercetin together in combination, we have seen inhibitory influence. So all this data together is implying that combination of methamphetamine and quercetin in vivo have shown a reduction in phage, which is then implicating perhaps DA oxidative metabolism, which is involved in the phage formation. And now to conclude, we can say that we were successful for a synthesis of the standard calibrator, which was for us fAGES-BSA. And then also that phage concentration is associated with the cellular localization. The most important outcome of this research was that phage concentration in vivo is independent of overall DL pools, but it is heavily influenced by DA impairment in transport and signaling. And also that combination of methamphetamine and quercetin together in vivo have shown reduction in phage formation, which is then implicated the dopamine oxidative metabolism involvement in the phage formation. And finally, these results presented here, we believe that can be used for further testing of phage in the context of the neurodegenerative disease disorder biomarker. And for the end of my presentation, I would like to thank my PI, Razi in whose lab I'm working right now as a postdoc. Also our funding, which is Croatian Science Foundation, University of Rijeka support, and our teammates or students which were working with us. That would be a bachelor Erasmus student, Katarina Jovic, and Emanuel Pištan, from whom I'm really super proud because he is only a high school student and he is able to follow up with this heavy and really complicated situation regarding phage formation in living organism such as Drosophila. Thank you. And yeah, I'm happy to take any question.
Thank you, Anna. It was great. And I see the implications for the neurodegenerative diseases. Probably you can expand on that a bit, unless we have further questions coming for you. So are there any questions for you? Are there any questions for Anna?
Nobody likes flies.
No, I think people like, but I think it means that it was very clear. and concise, and there were no questions. Yeah. So what are and this is also much out of my comfort zone as well. I work with humans at the macro scale level. But I do a lot of work in neurodegenerative diseases at the whole brain level. So I see where you're going. So what do you think? It's something which can be used. What are the translational implications here?
Well, for sure. I mean, dopamine is influencing the glucose levels for sure. And we know that if we are reducing or just playing around with the overall pool DA concentration, we don't see nothing. So that means that in normal health people, if you're I don't know, hungry, or if you're something like that, and you get your primary reward in a way of the food or something, then the phage concentration will be always remain the same. But for some reason, which I really now cannot elaborate good enough, because I don't know nothing about the glucose levels in these flies in their brains, and also nothing about the metabolism switching when we have impairment in DA. So for sure, there are some implications that we have something, I have some slides which are hidden for sure in this presentation. So we did some additional testing in vitro, not only in vivo, to play around with the protein structure and so on. And what we saw is that, for instance, dopamine is really tricky, although small, but really tricky compound, and it can bind to protein structure, but not through the amine group, but to the sulfur group, so thiol group in protein structure. So we believe perhaps that in that case, we are missing the modification which can be induced by the dopamine. Rather, there is a modification, but it's not glycation modification. So
yeah, that makes sense. I mean, perfect sense to me. I think we have Taha here, and I think he's here because I think we are getting late now. So we are over time, but we should thank Taha for his support for this session. He's at the back end, providing the IT stuff. Thanks a lot, Taha, and thanks everyone. Thanks all the speakers. It was great fun. It was very